Well, hello there. I wanted to make you a short video explaining the transformation procedure just a little bit, the theory behind it, and the predictions that you might have from this experiment. All right, so first to walk through the procedure, what you do is you take a small tube, a really small test tube. It's a little plastic sterile test tube. It holds about a couple of mils of liquid. And you put some transformation solution in it. And then you add some fresh cells to it, whatever the cells are that you want to transform. And the reason that you do this is because transformation happens in the wild, but not very efficiently. And so in the lab, we want to kind of push it to the max that we can get. And this solution is a salt solution that's designed to destabilize the membrane a little bit. So you put cells in a special solution, and then you add whatever DNA you want to transform into the cells. And then usually this is a plasmid, and one of the things that'll be on it is a gene for antibiotic resistance. That's a way that you can judge whether or not the DNA actually gets into the cell, because then you can later select for expression of that gene. Then you sit this solution full of cells and DNA in this liquid, you sit it on ice for about 10 minutes. And what's happening in that 10 minutes is that the DNA is coating the outside of the cell wall. Then the really important step is the heat shock step. And in the heat shock step, you put the cells, in this case, since it's usually E. coli, their favorite temperature is body temperature, which is 37 degrees. You put them at a heat shock temperature of 42. Now you might not think, oh, 42, it's like not that much higher than 37. But think about your own body at 42. That's a high fever. And over a prolonged period of time, that will kill you. And the same is true for E. coli. They can't survive this heat shock for long, so you put it at a very brief time. And the point of it is to destabilize the membrane. So you've already put it in the special salt that's destabilizing the membrane, and then this heat is intended to make the membrane very liquidy, so that the DNA that's accumulated on the surface can sneak inside. Remember that this DNA molecule is enormous and is charged and would normally never get past the membrane. So you ice it to let the DNA settle, you heat shock it to see if some of those DNA molecules can sneak inside, and then, because you did this brief heat treatment that for too long could be lethal, you stick it back on ice to let it recover, and then you feed it. You give it some broth, and you give it some basically broth that it likes that's food. After it's recovered, hopefully all the cells that you put in there in the beginning, in the first step right here, right here, this first step, hopefully those cells are still alive. And hopefully some of them have received the DNA. Not all of them will, because even with all of these interventions, it's not that common of an event. Then to figure out who's got the DNA, you take some of this liquid and you spread it out on a Petri dish that has a particular set of nutrients and maybe some challenge reagents like antibiotics on it. In these experiments, typically you'll do a negative control and then the real experiment. In the negative control, you do everything exactly the same, except you don't add any DNA. So when you do this negative control, you test it on a couple of media. So first you put it on LB, and LB is like a rich medium. It has no selection. It's not a selective medium. It doesn't kill anything. It's just lots and lots of good food. And the reason you do this is you spread a fraction of your negative control that was treated exactly the same as the transformation reaction on this rich medium to make sure that when you did the heat shock, you didn't kill everything. You take the same negative control and you put it onto a plate that has the same nutrients, but it also has the antibiotic added to it that these cells are normally sensitive to. And the reason you do this is that you're checking the cells are in fact really killed by this antibiotic. They should all be dead because none of them received any DNA that had an antibiotic resistance gene. With the actual transformation reaction, the one that has DNA in it, you would put that onto a plate that has the antibiotic. In this case, in the experiment that we're going to talk about in this lab, it's ampicillin. So we would add the antibiotic to the plate, and then what happens is that out of the thousands and thousands of cells that were put into the transformation solution in the beginning, only the ones that actually got the DNA and therefore received an antibiotic resistance gene will survive. It could be a really small number. And each one of those cells is going to grow into a colony. And that's what this plate will tell you. How well did that happen? Now, there might be other things on the plasmid that you're curious about, and in this experiment, we're curious about the expression of a gene called the green fluorescent protein, or GFP, which is regulated. Unlike antibiotic resistance, which is constitutive, GFP is regulated, and it is regulated by the arabinose operon. So we might add different nutrients to test whether this gene is being regulated the way we think it will. So in this experiment, the plasmid that we're adding is pGlow. I'm going to show you what that looks like. So one of the key features on pGlow, remember that plasmids, like pGlow is a plasmid, plasmids have only maybe just a handful of genes. Very small circular piece of DNA, like a mini-me version of the chromosome. And so one of the really important genes on this plasmid is called beta-lactamase. Here's its promoter. Here's the actual protein coding sequence. It codes for an enzyme called beta-lactamase. And what this does is it chops up penicillin and ampicillin and all the cillins. So it protects cells from, let's say, ampicillin. And since this is going into E. coli, which is normally ampicillin sensitive, this gene, if you have it, will allow you to grow even in the presence of ampicillin. So this gene is under a constitutive promoter, so if you get the plasmid, you will always be resistant. Now there's a few other things on this plasmid that are interesting. This plasmid is originally derived from the arabinose operon, so it still has the arabinose repressor encoded by this gene ARAC, which is also under a constitutive promoter. So if you have the plasmid, you've got the arabinose repressor. The rest of this regulatory module is the remaining regulatory region of the arabinose operon. Here's the cap binding site, there's the promoter, there's the operator. And instead of the arabinose metabolism genes, because those have been removed, we have engineered this with GFP, green fluorescent protein. So I want to show you the original arabinose operon so you kind of see where this came from and how it works. So the original arabinose operon looks like this. It's got these 3 genes for metabolizing arabinose under the control of a single promoter, and in between the promoter and the genes is an operator, and the repressor that's binding here is encoded by the ARAC gene over here under a constitutive promoter. There's also a cap binding site, so that when glucose runs out, cap will sit here and help to promote transcription. So let's just remind ourselves of what would happen in different kinds of sugar scenarios for this operon. If you were to feed cells with glucose only, then the ATP levels would be high, cyclic AMP levels consequently would be low, and so cap would not be active, so you wouldn't have anything here. And because cap is required to actually make this promoter, this crappy promoter, visible, we also don't get any RNA polymerase. Meanwhile, because there's no arabinose, this arabinose repressor is active and it's bound to the operator. So for 2 reasons, you can't get any transcription. So these genes are not expressed. If we switch to a different scenario and say, what happens if you just feed the cells arabinose? Well, what's going to happen first is that the repressor, which is sensitive to arabinose, is going to be disabled. So we don't have any blocks here. And furthermore, because ATP is low, because there's no glucose, then cyclic AMP is high, and this activates cap, and cap sits down here and makes this promoter visible so RNA polymerase can find it, and voila, we have transcription. And last but not least, if you put both sugars together, you get this wackadoodle situation where even though the repressor has been disabled, so it's not here, because glucose is around, ATP is high, cyclic AMP is low, so cap is not active. So it's not here to light up the promoter, so RNA polymerase can't sit down here, and so we don't get any transcription. Even though there isn't a negative block, we still don't get transcription because we don't have a positive push. All right, So what about this scenario? Here is our pGLO plasmid, where this is basically the exact same regulatory region, except that we've put GFP here instead of these metabolism genes. So in the event that we take our transformation solution and put it onto a plate that contains ampicillin and glucose, then what's going to happen? Well, glucose is present, so ATP is high, so cyclic AMP is low, so CAP is not here. Meanwhile, additionally, because there's no arabinose, this repressor is active. Now, if we add arabinose but no glucose, we get the relief of this repression. So the repressor is disabled, and because there's no glucose, ATP is low, cyclic AMP is high, CAP is active, it comes and sits down, it brings RNA polymerase, and now we can finally get transcription. And just to remind you, what happens when both sugars are present is that, when both sugars are present, then again, we relieve the repression of the repressor, so arabinose gets rid of the repressor, but because glucose is around, there's no CAP here. So no RNA polymerase, so no transcription. Okay. So what does this mean in terms of what we're expecting to happen when we take our negative control or our transformation reaction and put it on the plates that the experiment tells us to put them on? So let's start with the no DNA control. This is, again, a tube filled with cells and transformation solution, but no DNA, that's been subjected to heat shock. And now we're going to take it and we're going to put it on LB just to make sure that it's still alive. Okay, and if it is, all those in that, when you inoculate the cells in, you put in like tens of thousands, maybe hundreds of thousands. And so because there's so many, if they survived, you should see a lawn. Now you'll also take the negative control and put it on the LB ampicillin selective plate, and there should be no growth at all because these cells are naturally sensitive to ampicillin and we haven't added any DNA. Now let's take the actual transformation reaction. Again, remember the same number of cells was put in here, tens or maybe hundreds of thousands, but we did add DNA, and a very small subset of cells should receive this DNA. And one of the things that will happen when they receive it is that they will become ampicillin resistant. So on these three plates, let's start with the LB ampicillin. What we should see is the number of cells that got the DNA. Let's say it was 10 out of 10,000 plus. Okay, out of all these original cells, we got 10 that actually got DNA. And so every single one of those cells should form a colony, and so 10 would be the number that we'd see on this plate. And when we look at these other plates that have ampicillin, right, and ampicillin is the thing that determines, that is selecting for the cells that have the DNA, some of which have different sugar combinations. Would you expect the number of colonies to be the same or different? Hopefully, you think it'll be the same because what's determining how many cells there are growing is ampicillin, and that's present in all these plates. There's going to be a little bit of variation because of just random differences, right? This is 10, this is 12, this is 8, I think. And those are all within sort of standard error. Now the next question is, which of these plates will glow under a UV light? Green fluorescent protein, which can be expressed by this pGLO plasmid, is only going to be expressed sometimes, and to see it you have to shine a UV light on the plates, but nonetheless the question is, if you do that, who's going to glow? And remember our predictions, right? Our prediction would tell us that it would be this one where there's arabinose only and no glucose, because glucose would inhibit the expression of GFP. So in the instructions, you're going to look at some real data, and then you're going to calculate a transformation efficiency. And transformation efficiency is the number of transformation events per input DNA. Okay, so to figure out the number of transformations, we have to count colonies, and if we have put a transformation reaction on multiple plates, we could average over those different plates to get a more accurate number. And we also need to know the amount of DNA per plate. which we can calculate if we know the volume of DNA we used and the concentration of the solution. And then we just plug those numbers in. We put colonies up top, or our average colonies, divided by the micrograms of DNA per plate, and voila, we get our transformation efficiency in CFUs per microgram DNA. And that's the little intro. So you can go ahead and read the instructions and get started on the lab.